It is our remarkable brains that endow us with mobility and curiosity. They power our thoughts and actions and are responsible for everything we sense, feel, discover, and remember.
The brain is what allows us to make sense of the world.
And perhaps most importantly, our brains give us the ability to understand, create, and do remarkable things. But exactly how our brains give us any of these abilities has so far defied scientific explanation.
With brains, we know next to nothing. And that makes it quite different, actually, from other organ systems, where no one would say we know next to nothing about the heart. We can replace a person's heart with an artificial heart.
In physics, we've been able to not only understand what atoms are made of, we also know how galaxies, millions of light years away, behave. So how is it possible that we can understand the universe better than we can understand the brains that we use to figure out the universe?
But we may finally be on a pathway for understanding our brains in ways that have eluded scientists and philosophers for centuries. Could a comprehensive map of the vast network of brain cells called neurons become the starting point for unlocking the mysteries of the brain? We're not totally in the dark about how our brains work. By illuminating brain activity, MRIs and other imaging systems have enabled us to identify regions of the brain that process sensory information, like smell, vision, and touch, that exert motor control, that solve problems, or process music, language, and much, much more. But what we can't see on MRIs are the super-thin neuron networks that facilitate all our thoughts and actions. Individual neurons in these networks are so slender, you can only see them under much higher resolution. Only then do their unique shapes reveal how specialized they are among all the cells in the body. Neurons receive information in the form of chemoelectrical impulses through their root-like branches called dendrites, and they send that information via axons to other neurons. So neurons are the building blocks of a remarkable communication system threading throughout our bodies and centered in our brains.
So each cell is sort of like part of a Twitter universe, if you will. It's receiving lots of information on its dendrites, and then from that information, it decides what is newsworthy, and then it sends that information out to all of its listeners. And that's true of every cell of the hundreds and hundreds of millions of cells in the brain.
And this vast network of neuronal cells is connected by trillions of cellular junction points called synapses.
The synapse. It's a place where an axon releases a chemical called a neurotransmitter that activates a receptor set on the dendrite of another nerve cell.
Synaptic connections can weaken or strengthen over time. So besides relaying and processing information, some experts believe they may also store information, like our memories.
Memories are probably stored in the patterns of synapses that were activated at the time they were formed. Now, not all memories are necessarily stored. And in fact, it's important that not everything is stored. Otherwise, your brain will get filled up.
It seems like our brains are already filled up. They contain billions of thread-like neurons, so many that if we placed each strand end to end...
But it looks like it would go from here to the moon and possibly back, but I'm not sure about the back part.
And this neuron jungle is so dense and crowded, it's almost impossible to pinpoint the neuronal networks associated with a given thought or action. And this lack of knowledge has been a major stumbling block for decoding the brain. It's like our brain is a map, where we know the location of language and music cities, motor control, and other towns, but can't see the roads that connect them, or even the roads within and around them.
And so the idea that we're continuing to try to wade through the brain to understand how these networks are really processing information without even this map seems like it's not the most efficient way to proceed.
That's why many neuroscientists believe if we really want to figure out the brain, we need to start by creating a kind of neuronal roadmap called a connectome.
So this incredible kind of spider's web of how every neuron connects to every other neuron, if you mapped it, that would be the connectome.
But with billions of cells in this endless forest, how does one even begin?
To actually map the brain requires something at like nanoscale imaging.
Which means starting with thin slices of a brain and imaging them with an electron microscope. In certain operative procedures, tiny pieces of discarded brain tissue can be collected for analysis. But a human brain is so large and its neural network so vast, attempting a human brain connectome is for now a bridge too far. So researchers are starting with the brains of mice and other animals to see if they can make connectomes and what they might learn from them. But even small brains are huge challenges.
Animals like the fly has about 1 million neurons. The mouse has about 100 million neurons.
So is creating any connectome even possible?
Truth be told, it's very hard to get a connectome. In fact, there are only a few connectomes that have ever been gotten, and mostly of teeny-weeny little animals like worms that only have 300 nerve cells.
This tiny nematode worm is called C. elegans. In the 1970s, Cambridge University molecular biologists John White and Sidney Brenner tried a nematode experiment.
Sidney Brenner came up with the idea that we needed a connectome to understand how the brain worked. So he wisely picked the simplest brain, the brain of a worm, C. elegans, which only has 302 neurons.
The Cambridge team thinly sliced the tiny worms and photographed each slice using a film camera mounted on an electron microscope. They then printed out large photographs and used colored pens to trace the neuron pieces they saw in each slice until they had every neuron mapped. It took nearly a decade, but in 1986, C. elegans, with its mere 302 neurons, became the first and to this day only organism with a fully mapped connectome.
C. elegans was great, but C. elegans is not highly intellectually capable, shall we say. Its brain is simple. Even the mouse brain has 10,000 times as many neurons. So we face this dilemma. The really capable animals that we all want to understand, we don't have anywhere near the technical capabilities to tackle that.
Think about what would 300 million cells require. Instead of 300 cells, it would require 1 million times longer. That would be 10 million years. So nobody would do that experiment.
We quickly realized that the only way this was possible is if we could automate every one of these processes. Automate how we take the images, automate algorithms that automatically identify neurons and trace their connections for us. Essentially made the entire thing digital.
Over the last few decades, that's exactly what researchers did, working on mini neuronal maps from pieces of various animal brains. Although some processes vary, they begin with a tiny brain sample, that is stained with a metallic liquid, encased in plastic, and in most systems mounted on a chuck that moves up and down across a diamond knife that slices it. The super-thin slices go on a conveyor belt and ultimately to electron microscopes that image each slice with remarkable clarity. The images are fed to a computer that restacks the slices in the original sample, only now with the neurons clearly outlined. Another program isolates each neuron by color and begins to fully render them. Here we see the red dendrite of one neuron and the green axon of another. The computer will render all the neurons in the sample the same way.
There's something aesthetically very pleasing about this kind of data because it reveals something that you've known is there, but you never actually could see before.
But these neuron images from a mouse brain, created almost a decade ago by Jeff Lichtman's lab at Harvard, also presented a formidable reality check.
We completely rendered every single item in this teeny weeny little cylinder. It was just fantastically beautiful to see, and also so much more complicated than anything I had ever seen before. I despaired a little bit seeing how small the amount of progress you've actually made relative to big questions like doing a whole mouse brain's connectome.
If it wasn't clear before, it was now. Creating a full connectome for even a mouse-sized brain would require a massive effort. It was again time to think small. The brain of a Drosophila fruit fly is the size of a poppy seed on a bagel. Yet it contains about 100,000 neurons and millions of synaptic connections. But in 2020, researchers at the Howard Hughes Medical Institute's Janelia Research Campus announced they had mapped the fly's hemibrain. This is its outline, and these remarkable images show some of its 25,000 neurons. Although researchers still have more than half a brain to go, these partial maps together comprise the largest connectome ever produced.
Seeing the connectome for the first time was just absolutely breathtaking. You at once feel hope that you can actually see the reality of this complexity, and maybe as a scientist, a little bit of worry or despair at how challenging it still is just to unravel the function of that mass, even for a fruit fly.
We're starting next Wednesday with the program.
Reaching this milestone has been the dream of the project's founder and guiding spirit, Jerry Rubin.
I had to convince the people in my organization that this was worth doing. And it was a 10-year investment of tens of millions of dollars. So I lost a lot of sleep, like, I'm going to look really bad if this fails.
The Janelia Fly team developed new methods for preparing samples and automating the system. They began using focused ion beam microscopes. These can whisk off and image the nano-thin layers almost simultaneously. The machines operate automatically and continuously as long as samples are replenished. And all this has speeded things up considerably.
Everything is being done by computer instruction. Every time humans get involved, it slows things down.
Although Janelia's machines have turned out hundreds of thousands of 2D images, to render them into three-dimensional neurons required massive amounts of data processing. So Janelia went to a world leader in data processing for help. Google not only agreed to partner with Janelia, the company decided to do the computational work for free. The obvious question, what motivated Google to make such a commitment?
This was an area in which advanced computer science could really make a difference. So, the idea of taking these very large three-dimensional imaging data sets, reconstructing them, annotating them, this is the type of problem that Google had developed expertise in, through working on things like Google Maps and Google Earth. It presented new challenges, and, we want to make computers smarter.
Google needed to adapt algorithms used for object and pattern recognition to see if these could recognize neurons. They began by feeding their machines precise neuronal images supplied by Janelia so the machines could train themselves on what to look for. The machines also rely on artificial neural networks that mimic the way neuroscientists believe the brain processes visual recognition in animals like these mice.
And what they found was that the brain had one layer of cells, which they called simple cells, and these would respond to oriented gratings of light. And then you had another layer of neurons, which they also discovered in the brain of these animals, called complex cells. And as long as one of the simple cells responds to this feature, then complex cells respond also. And then you can combine these layers to get progressively more complex and higher level representations of any image.
Machine models of these visual processing systems are called convolutional neural networks, and they are today a cornerstone of artificial intelligence.
Even something like a self-driving car, which might have a camera in the front of it and is looking at a visual scene and trying to figure out where's the road, where's the stop sign, is there a traffic light? Those decisions are often being made by some type of convolutional neural network.
But neurons have extremely complex shapes, so Viren Jain had to create an equally complex version of Google's recognition algorithm.
You pick a point in the image, let's say any point, and the algorithm starts trying to figure out what are the boundaries of the object that was associated with that point that you picked. So from that point, the algorithm is looking around and deciding what else is part of that object. So what you're seeing as that point moves around is the algorithm progressively expanding its field of view. So it's starting in just one place, and then it's moving over here and saying, okay, well, what else is part of this object? And so it's expanding the shape as it goes along.
Although the system is extraordinary, it can still make mistakes.
The computer analysis is great, but it's not perfect. So what we need to do is review it with humans. And the humans look at it and they say, oh, these things are joined together and they shouldn't be together. We have a Frankenbody, we like to call them.
In this Frankenbody, the computer sees one neuron, but it's really two. It took scores of Janelia proofreaders years to correct the mistakes. But as they did, the new information went back to Google, and its algorithms kept getting better. Jerry Rubin now says it should take about a year or two to completely finish the fly Connectome, which would come in under his original estimate. But despite the coming milestone, this is, after all, a fly brain we're talking about. What could its Connectome possibly tell us about the human brain?
Well, it turns out that fruit fly brains are made of exactly the same components that our brains are, these neurons. And they actually talk to each other in the same ways, exchanging chemicals and exciting one another, as it were, to pass on information. So if we can understand the building blocks of that communication in a fly's brain, that applies to our brains just as well.
Gwyneth Card leads a Janelia team that experiments with fruit flies to try to understand how their brains react to things in their environment that motivate behavior.
We try to study really fundamental behaviors that our brains all evolved to be able to do. So these are things like eating, sleeping, or in the case of my lab in particular, how to not be eaten. We're interested in how a fruit fly evades being eaten by damselflies or other predators that try to capture them. What we learned from our research is that even flies are carefully considering a strategy for escape. It can do a faster takeoff, where it doesn't raise its wings, it just jumps off the ground. Or it can do a slower kind of takeoff, where it does raise its wings and then slowly depress them and get off the ground in a longer time. And we understand now the neurons involved in the choice between those kinds of takeoffs.
Flies aren't the only creatures with quick escape abilities. But we don't know which neurons in our brains message such actions, whereas Gwyneth Card's fly team can use imaging techniques and the Connectome to identify the neural network controlling escape behavior.
What the Connectome allowed us to do is it allowed us to go beyond that one neuron that's involved and say, what is actually the network here? How is that neuron getting information? What kind of information is it getting? What is the animal really actually trying to understand about the world? And that was virtually impossible to do in a comprehensive and systematic way without the Connectome.
And besides mapping and tracing neurons, Janelia's Connectome even pinpoints synapses. Seen here as microscopic dark smudges in the red circles that link neural networks. And this is helping other neuroscientists as well.
They have revolutionized the thinking of how circuits work in the fly brain. It's no longer modeling it. You actually know who's connected to whom. Once you know who's connected to whom, you no longer have to try to guess.
In a relatively short time, you've seen people really not only take advantage of the hemibrain data set, it's also having a broader effect in neuroscience of showing that this type of information about the nervous system is really quite fundamental and can be extremely valuable.
Success in building a fly connectome has energized ongoing efforts by several research groups to pursue a far more complicated mouse brain connectome. The Allen Institute for Brain Science in Seattle has been a major contributor, and in 2019 announced that after nearly a decade of work, it had imaged 100,000 neurons and 1 billion synaptic connections in a cubic millimeter of a mouse brain. It was a significant success, but dauntingly, there were still 499 cubic millimeters left to map. As several research groups travel the long pathway to a mouse connectome, others are pursuing an interim step.
Between the mammalian brain of a mouse and an invertebrate, like Drosophila or C. elegans, there's a whole group of animals that have backbones about which Connectomics could provide a lot of insight. And perhaps no animal is better suited for this than the larval zebrafish.
The zebrafish we see in tanks are adults. Connectomic researchers are more interested in their babies.
Zebrafish that I work with, you will need pretty much a microscope to look at.
Mariela Petkova is a postdoctorate neuroscientist at Harvard University who uses Connectomics to study the brains of zebrafish.
It is surprisingly similar to us. We share a lot of genes. We have shared a common ancestor. I think about 400 million years ago, we both were swimming around. They start as a single cell, and 7 days later, they have 100,000 neurons, and in 3 days, it already starts swimming. Imagine, in only a week, you have to build a circuit that hunts, that can take in the information around it, and build a machine which can do such a complicated action.
And because they are transparent, using a microscope, she can see the neuronal pathways involved in actions like tail flicks, and even measure them.
You can put in a molecule which tells you when the neuron turns on, and see the molecules light up, almost like a light bulb.
And the connectome she is creating can help pinpoint the flick neurons precisely. To create their connectome, the lab uses the basic slicing and imaging procedures, and here, too, Google helps process the images. But Mariella's lab has a unique community of checkers.
We bring in a lot of people to proofread. We bring in college students, we bring in postdocs, graduate students, professors. We also bring in high school students, and I brought in my mom. My mom does all the work from Bulgaria. This is something very exciting about Connectomics. You can do all the work from your computer, and you can be anywhere in the world. Connectomics has a huge potential for engagement from the public.
In a popular interactive game called iWire, developed by the Sung Lab at Princeton, players from all over the world vie with each other to see who can best reconstruct neurons in a mouse retina. The site also teaches gamers about the intricacies of neurons and synapses, and iWire could clearly become a training ground for citizen proofreaders, which would certainly help with the massive task of completing a mouse connectome. Although a mouse brain is 1,000 times smaller and far less complicated than a human brain, as mammals, their brains will be fundamentally like our own, as are many of their biological systems.
The mouse is probably the most popular animal for study in biology. Not the least of which is diseases that have correlates to human disease are being made in mice all the time. So there are cardiovascular, diabetic, Alzheimer's, mice, to name a few, and many of these are neurological diseases, like Rett syndrome and autism spectrum disorder. These animals provide us with an opportunity to look at the brains of animals that have cognitive problems that are similar to diseases that humans have.
Because mouse brains are closer to ours, short of a human brain, right now, their connectome would be the ultimate prize. But to do this, the costs for data processing alone would be, well, mind-boggling.
If you took all the material that lives in the Library of Congress, not just the books, but all the audio, all the video, every movie, every song that has ever been written, and you piled it all together and said, how much data is that? And then you asked how much data a mouse brain would be. A mouse brain might be something like 100 to 1,000 times more data than has ever been collected altogether in the history of humanity. That would be an amount of data called an exabyte of data, which is 1,000 petabytes of data, which is 1 million terabytes of data, which is 1 billion gigabytes of data. If we really want to map a mouse brain, neuroscience is going to have to embrace big science.
There have been in the history of science many very large projects. Going to the moon is a good example of an insanely large project.
Contact light. Okay, engine stop. We copy you down, Eagle.
That when it was proposed, I think people thought this was perhaps too challenging. The human genome was another project that was considered extraordinarily hard.
And some doubted it could even be done. But the Human Genome Project was completed successfully and led to key breakthroughs in medicine, anthropology, and forensic science.
So we're trying to do the same thing here with doing the connectome. It isn't the knowledge itself, it's an infrastructure, it's a foundation, it's an enabling tool that gets us there. You can't imagine trying to understand the brain without this.
When I started in this field 50 years ago, there was no hope of, doing this. It was just barely beginning to get started with manually tracing the C. elegans. And if you look at the fly brain, even a quarter of that has revolutionized the thinking about circuit analysis in the fly brain. So the same thing will happen in the mammalian brain, in the human brain, that the more we know, the better we can understand what's gone wrong in disease processes, what's gone right in genius.
I think all we need to do a mouse brain is the will to do it. We have the technology. We have the people who have this knowledge. It's a huge undertaking. And I think we just have to decide as a community and that it's worth it, that this is the information we need in order to advance the frontier of brain science.